Hi guys, Miss Francis here. This morning what I'd like to do is compare replication and transcription and then compare transcription and translation as a nice wrap up to all three of these processes that play a vital role in protein synthesis. So let's first look at the replication side of the Venn diagram. Within DNA replication, we know that helicase is going to unwind our DNA strand. Now, before that DNA strand can even be unwound, we know that replication occurs during the S phase of interphase. We also know that replication involves the separation of sister chromatids. Okay, so helicase is what unwinds the DNA. SSBP, single strand binding proteins, are what holds that DNA open or unwound. You've got one side is a leading strand, one side is a lagging strand. The leading strand builds DNA continuously. The lagging strand builds it in fragments known as Okazaki fragments. That Okazaki fragment is then glued to the neighboring Okazaki fragment by ligase. Now, DNA polymerase is the enzyme that actually adds those nucleotides onto the leading and lagging strand. Well, how does DNA polymerase know where to start? You've got this RNA primer that is a section of RNA that's going to tell the DNA polymerase where to start its replication. Do you notice something on this side? RNA polymerase. Is RNA polymerase correctly placed? Nope. RNA polymerase is involved in transcription. Now let's take a look at transcription. Well, during transcription, you've got an initiation phase, an elongation phase, and a termination phase. And it's this RNA polymerase that does transcription. Now, how does transcription actually start? It involves the Tata box. It involves your transcription factors binding to that Tata box. Let's take a look at this center. Do you notice that both DNA replication and transcription occurs in the nucleus? So before the messenger RNA that gets made during transcription can leave that nucleus, it needs to have a cap and a poly-A tail added on either end as protection, because you've got hydrolytic enzymes in the cytoplasm that surrounds the nucleus. So you want to give that messenger RNA some protection once it leaves the nucleus. Now, once that messenger RNA is in the cytoplasm, you have these introns that are going to be removed. Introns are that non-coding area found in your DNA. And then the exons are the coding sections of your genes that are found in your DNA that will be expressed. Both of these processes use a DNA template. Replication uses that DNA template to create a new strand of DNA, and transcription uses that DNA template to transcribe DNA into messenger. So now let's compare transcription and translation. Transcription is taking DNA and transcribing it into messenger RNA, where RNA polymerase is the enzyme that's going to add those nucleotides to our DNA strand. Transcription starts when the transcription factors bind to a Tata box, and this whole process takes place in the nucleus. Do you notice that I have put rRNA on this side? rRNA stands for ribosomal RNA. Guess what? Ribosomal RNA is involved in translation, not transcription. rRNA will be found at the ribosomes. During both processes of transcription and translation, you've got three stages: initiation, elongation, and termination. During translation, you have transfer RNAs acting as taxis, and those taxis are carrying with them amino acids. Well, how does each taxi know where it needs to go? Because that transfer RNA, that taxi, contains an anticodon that's going to match to the codon that's found on the messenger RNA. Methionine is the amino acid that's attached to the start codon. The termination sequence tells us that we can stop doing translation. Our amino acids are going to be able to form a chain, a polypeptide, using peptide bonds between each one of those amino acids. And chaperones are going to help that protein build into its, or fold into its appropriate structure. Now, I can actually prevent translation from happening or stop translation as a method of gene regulation using what's called siRNAs. So I hope you guys found that comparison of replication and transcription versus transcription and translation helpful. What I would do is write out the steps of replication, write out the steps of transcription, write out the steps of translation, and then compare and contrast those lists. That's another way of making sure that you know the steps of replication, the steps of transcription, and the steps of translation, because each one of those processes plays a critical role in creating the proteins, which then are life. We can't live without making those proteins.